Hi everyone, my name is Brett Drummond. I'm an MS researcher, science communicator, and co-founder of MStranslate. I'm here today to talk to you about another research paper that's just been published in multiple sclerosis. And today's paper's got to do with the gut microbiome, which I know is a topic that's really interesting to everyone. So let's get into it. Okay, so as I said, today we're going to be looking at a newly published research study looking at the gut microbiome and its role in multiple sclerosis. So the study in question is this one, recently published in Science Immunology, and it's called IL-17 Controls Central Nervous System Autoimmunity Through the Intestinal Microbiome. Now again, that's quite complicated. We don't need to understand all of that, but what I will say before we get into talking about the rest of the paper is just commenting on this molecule here called IL-17. So IL-17 is essentially a chemical messenger that's involved in communicating with the immune system. It's what we call a cytokine. And IL-17 has been shown to be involved in multiple sclerosis and other autoimmune diseases through their interaction with a specific type of cell in the immune system called Th17 cells. So IL-17 and Th17 cells have been shown to play a role in multiple sclerosis, and so that's where the interest is in terms of looking at this in more detail. So if we go into what they did in the study, essentially what they did to start with was that they took mice, so this is an animal study, and they were interested in looking at the mouse model of multiple sclerosis, which is known as EAE, or experimental autoimmune encephalomyelitis. So they took specific mice that had been genetically modified so that they couldn't express that chemical messenger, the IL-17. So essentially these are normal mice other than the fact that they don't produce any of that IL-17. And what they saw when they tried to induce the disease in these mice, they tried to have them develop EAE, is that actually these mice were less susceptible to disease than mice that could make IL-17. Now, they weren't completely protected. Some mice still did develop a disease. It was slightly less than 50% still did develop a disease. But even in these mice, they saw that the onset of the disease was delayed. It took them longer to develop the disease. And also they didn't have anywhere near as bad a disease experience. Their symptoms were quite mild. And interestingly, when they looked into that, the first assumption might have been that the immune system was slightly different in these mice because of the fact that they couldn't express IL-17. But they didn't see any differences in terms of the composition of the immune system between these mice and the control mice, the normal mice. So they wanted to look into that a little bit further, and when they did, what they found was one of the biggest differences was that there was a change in the gut microbiome in these mice compared to mice that could express IL-17. And so here is this first piece of evidence that's suggesting that this molecule that we know does play some role in multiple sclerosis actually plays a role in shaping the gut microbiome as well. So they wanted to investigate that further. And so what they did was that they co-housed, so put in the same cage, put in the same environment, these mice that couldn't express IL-17, shown here on the left, and normal mice, what they called wild-type mice, shown here on the right. And they did a couple of different things, but I'm going to focus on one particular experiment of that, where they did fecal transplants. And we know that fecal transplants are a way of being able to shape the gut microbiome. So essentially the mice were given antibiotics to wipe out the gut microbiome that they already had, and then they were given a fecal transplant from the other mouse to see whether or not they could then colonize that mouse's gut microbiome, and if so, what impact that would have on the disease. So we're going to look at this in both directions. So firstly, they took a fecal transplant using fecal matter from the normal mice, the mice on the right, and they put that into the IL-17 deficient mice. And what they saw when they did that was that now these mice were completely susceptible to developing the disease. So remember, before, in that first experiment, they seemed to be somewhat protected. Much less of them developed disease, and the disease that they got was mild. Once they got the gut microbiome from these normal mice, that disappeared, and they experienced the disease in the same way. So the researchers were then interested in going, well, could this go the other way as well? what if we take the gut microbiome from these IL-17 deficient mice, the ones on the left, the ones that seem to be protected, and transfer that into normal mice? Would they then also be somewhat protected from developing disease? So they did this experiment, and what they saw was that that wasn't the case. They didn't have any protection from EAE, and they developed the disease normally. And so from that, what they concluded was that the only way that you could get that gut microbiome was in the absence of IL-17, and so it couldn't exist in these normal mice because they did have that IL-17, that chemical messenger there. And so they then went on to look at, well, what happens if we just take these mice and have them express IL-17 specifically in their gut? So this mice on the right now has been genetically engineered again, so that everywhere else in its body, it doesn't express any IL-17, it can't make IL-17 there, but in its gut it can. What would we see in that instance? And what we saw, or what the researchers saw, was that those mice were susceptible to EAE, so they developed disease normally, and the gut microbiome had changed to be more like a normal mouse. And so from this, they were able to conclude that it's specifically the expression of this chemical messenger, the IL-17, that cytokine in the gut, that's important in shaping the gut microbiome in a way that makes it susceptible to disease. And if that was absent, they developed a different gut microbiome that seemed to protect them, at least in this animal form of the disease. So there are a number of questions that still need to be answered based on this, because as we know, this is just an animal model, and so it doesn't necessarily mean that this will translate across to human disease, but it provides a really interesting link in terms of how the immune system and messengers of the immune system may be linked to the gut microbiome and how that may be involved in multiple sclerosis. So as always, if you do have any questions about this work, please do comment below the video and I'll make sure that I answer all of your questions as quickly as I can. And to ensure that you stay up to date with all of the new content that we're publishing, make sure you subscribe or follow our digital channels so that you can see our new content when it's published. Thanks everyone, I hope you found this video interesting and informative, and I look forward to bringing you another update soon. Bye.